Year 11's today's lesson is on eukaryotic organelles. Some of you have already covered this in class. We're going to try and do it via video today. So the first thing to remember about eukaryotic organelles, or eukaryotes in general, is that they dominate the current landscape. So they dominate the biological landscape. The reason for this are organelles. Organelles are one of the central reasons for that. Organelles provide a clearly defined location, so they increase organization. This leads to a remarkable level of specialization. This allows the cells to be very, very unique and very, very specific in what they do, so that we can drive a lot of the processes that we have in our bodies. It greatly improves efficiency and also allows for remarkable complexity in diverse multicellular organisms. So the making and handling of molecules. One of the problems as a heterotroph or as a eukaryotic organism is that we need to make our own organic molecules. So the big reason or the big drive behind that is so that we can create our own energy sources for photoautotrophs, or for organisms that can actually do that, or for heterotrophs so that we can convert the organic molecules we get from our prey or from our food into something more useful for us. One of the most important facilities, or one of the most important cellular mechanisms for that is the endomembrane system. Now, endomembrane system works a lot like the organ systems that we covered last semester. It is an organization of lots of these different organelles, these small organs, mini organs inside of cells that work together. And the endomembrane system is made up of ribosomes, endoplasmic reticulum, Golgi apparatus, vesicles, as well as a nucleus. The nucleus is the start of this whole process, and the vesicles are the end. In the next few slides, we're going to go through each of these in detail. The nucleus, it's a large organelle, made up of a nuclear envelope, which within the nuclear envelope is the nucleoplasm, the fluid within the nuclear envelope. The nuclear envelope is dotted with pores, and these nuclear pores allow the nucleus to import and export only certain molecules. So it allows for this selective exchange. The nucleus contains the organism's DNA, and this holds the code for synthesis of all organic molecules, proteins, nucleic acids, as well as the mechanisms for lipid synthesis. DNA is organized into genes, and in eukaryotes this is further organized into chromosomes, and these have specific sequences of genes. So we have our basic structure of a nucleus. Generally speaking, so this is our cell. Within the cell, we have a nucleus. And the nucleus itself is broken up into, or is broken by, these pores. So obviously in biological diagrams, we need to make sure we have our labels. So here is our cell membrane as the outer barrier. This is obviously an animal cell because it has no cell wall. Inside here, we have our nucleus, and then these red dots here are our nucleopores. Within the nucleus itself, so we'll draw that. Within the nucleus itself is our DNA. Organized into genes and chromosomes. So the nucleus, a large part of animal cells. It takes up about 30% of animal cells primarily because it's such a critical part. It's equally large in plant cells, but plant cells are obviously a lot larger than animal cells. Unless a cell is dividing, the DNA itself is unrecognizable, or it isn't easily differentiated from any other structure in the cell. Only when the cell is dividing is it easy to see the DNA itself. So we normally draw the DNA in a cell at rest as a kind of vague sort of murky soup. Within the nucleus, there is also a region called the nucleosome. And the nucleosome is where we make all these other parts. So not just the DNA, but we also synthesize ribosomal RNA, which we'll cover when we get to ribosomes. So ribosomes. Where are the ribosomes? Well, to talk about ribosomes, we need to add another organelle. So in our ribosome, we'll borrow green again. Just outside of our nucleus, or attached in what we suspect is attached to the nuclear envelope, is our endoplasmic reticulum. Now in this case, it's the rough endoplasmic reticulum. So that's the blue sort of zigzag, and then the green dots there are the ribosomes. And we also need to draw, and we'll draw it a little bit further away, we also need to draw a similar sort of structure, which is the smooth endoplasmic reticulum. Most often we shorthand this to lowercase r and then capital ER and a lowercase s and capital ER, the smooth endoplasmic reticulum, just because they're long and it's relatively boring. So the ribosomes were found mostly dotted throughout the rough endoplasmic reticulum. There are some free ribosomes, but there aren't terribly many. Now the ribosomes are an important structure. They're quite simple, but they are very important. And they're made up of two main bodies. They're made up of a large, so we need to give this a title. So this is our ribosome. And they're made up of a large subunit and a small subunit. Now, as the nucleus makes mRNA, which we'll get to a little bit later on, but the nucleus converts the DNA, converts the deoxyribonucleic acid to messenger RNA, which is a different type of nucleic acid, and this gets converted and brought out to the ribosomes. Now the ribosomes convert that sequence, convert that nucleic acid sequence into an amino acid sequence. So they're making the genetic information that we have into protein information. The nucleus talks in its own language, and the nucleus talks in the language of nucleic acids. Proteins, or the rest of the cell, talks in language of proteins, an amino acid language. And it's the ribosomes that translate between these two languages. The ribosomes convert the language of the nucleic acids, convert the language of the nucleus into the language that the rest of the cell understands. So it's at the ribosomes that we make our proteins. Where we're looking at the endoplasmic reticulum now. So the ribosomes predominantly dotted along the rough endoplasmic reticulum. The endoplasmic reticulum, as we've said, is made up of a smooth and rough ER. The smooth endoplasmic reticulum involved in synthesis of organic molecules, primarily lipids and fats. Anything that's non-protein based is going to go towards the smooth endoplasmic reticulum. The rough endoplasmic reticulum, because it's dotted with so many ribosomes, is made up of, or is going to focus on producing proteins. So these are very, very important structures within the cell. The rough ER also contains numerous enzymes that attach sugar molecules to proteins that allow it to make glycoproteins. Both of these structures, the rough and smooth endoplasmic reticulum, because they're part of the endomembrane system, they are very heavily membranous. They are very large membranes, which means that they have lots of surface area. They are complex, they are folded, they have lots of surface area. In the diagrams that we've got on the next slide, we've got the endoplasmic reticulum, the rough endoplasmic reticulum primarily shown as a series of folded stacks, or a series of folded sacks, sorry. If we're looking at, we'll get rid of our ribosome. So if we're looking at our rough ER, we've got our nucleus on the outside of it, so we'll just draw part of the nucleus, and then the rough ER sort of comes out of it, and they're all interconnected. It's difficult to draw in 3 dimensions, but they're all interconnected, and they're just dotted with these ribosomes. So we get our labeling. Each sac is called a cisternae. So the cisternaes, or there's plural of cisternae, the cisternae are stacks of these sacs, these rough endoplasmic reticulum sacs. And this is the site where protein synthesis occurs. We're moving now on to the Golgi apparatus. So we need our diagram to continue. And the Golgi apparatus, separate from the nuclear envelope, obviously these diagrams are not particularly to scale, the Golgi apparatus receives all of the organic molecules made by the cell. So this is our Golgi apparatus. And it receives these organic molecules from the cell. And it processes them and packages them. So it puts the final finishing touches on it. The Golgi apparatus, again, made up of a series of stacks. In fact, it looks, in a diagram, it will look like a series of stacked plates. That's the classic sort of description of it. The Golgi apparatus has two important roles. We've mentioned them very briefly earlier. It processes and it packages. Now, the processing sort of thing, if we imagine the Golgi apparatus like a bit of a postal service or a post office, it receives the goods and it might add little bits and pieces to them. So perhaps there's a mistake in the label and they need to repair the label. So it might do that. It doesn't necessarily commit any repair, but it's going to do all of that packaging, all of that final processing sort of stuff. But the most important thing for the final product is that the Golgi apparatus packages it for transport. And this is where our analogy of the post office works really, really well. The Golgi apparatus puts these molecules, these organic molecules, into little vesicles. So it actually buds off. These are, once again, these are folded sacs, and these molecules bud off of these folded sacs, and they go off into the cell, into vesicles. Now, the vesicles have to be labeled. They need to be labeled properly. They need to be labeled appropriately. The easiest way to do that is to attach proteins and sugars to the outside to communicate to the cell, which works in the language of proteins and sugars, what exactly is going on. So it does this in a variety of different ways. And it's beyond the scope of our course exactly how it does it, but you need to know that it uses these proteins to do that. The next thing we're going to look at now is we're going to look a little bit into mitochondria and chloroplasts. I want to, though, just quickly add to that diagram these vesicles. So the ribosomes produce their own vesicles, and then the Golgi apparatus is going to package those vesicles for transport. So we'll call the ones coming off of the Golgi apparatus transport vesicles. And that's going to either go off, out into the cell, or it's going off into the big wide world, or it's going to stay and remain stored in the cell. So these are our endomembrane organelles. The next set of organelles that we're going to be concerned with, and we're gradually running out of space here, are our energy organelles. Now we need to get rid of our cell here, and we need to start thinking about plant cells, because this is obviously an animal cell. So plants are able to convert light energy into chemical energy. Very basic sort of, or very simple sort of description of that is we can just use our chemical equation. So chemical equation, or the chemical equation, or what we describe that as, is photosynthesis. So the overall process, we call it photosynthesis. And we can simplify this in just that chemical equation. So we start with carbon dioxide and water, and we end up with glucose, which is a sugar, and oxygen. And we need to balance that, so we just put a 6 in front of the carbon dioxide, water, and oxygen, and we've got our nice balanced chemical equation. In order for this to happen in eukaryotic cells, however, because we're complex, because these cells are complex, they have their own organelles to do so. And these organelles are chloroplasts. Now, generally speaking, chloroplasts are just sort of drawn as a bit of an oblong sort of shape, and they will be full of these disc-like structures. I'm trying to draw this as large as I can. So be full of these disc-like structures. Now, this is again increasing surface area. These internal structures are cool. We've got a particular name for them. So this overall is our chloroplast. And the individual structures are called thylakoids. Because we are an organized creature, we as eukaryotes, we organize them into stacks. And we call these stacks granum. And it's sort of a basic sort of principle. Granum comes from the same sort of word as granary, which is where humans store our grain, store our food. So it's a similar sort of thing. This is an area where food is made. Now, photosynthesis uses the energy in sunlight, and it takes that energy, and there's a special protein called chlorophyll, which is stored in these chloroplasts that converts that sunlight into stored chemical energy. And the energy is stored between the carbon bonds of this glucose molecule. When we go into our next organelle, we look at mitochondria, we'll see exactly how important that is. So cells need energy. This photosynthesis, this process of photosynthesis with the chloroplast and all of these different complex structures really gives us an indication of how important energy is. Now, this is stored energy. Energy is stored in this glucose. So we can just put up here, this is our stored energy. But storing energy is no good unless you can get it out again. Now the principle here is exactly the reverse. When we're trying to get rid of our energy, we need to reverse this chemical equation. And we have, obviously, because we're multicellular organisms, because we're eukaryotes, we have our own organic molecule, or we have our own organelle to do that. The organelle you'll all be familiar with. It's the classic powerhouse of the cell. It is our mitochondria. But the process is called aerobic respiration. And you'll see what I was talking about with reversing the process. Aerobic respiration is the exact reverse of photosynthesis. Now, the analogy doesn't continue terribly far beyond that. The mitochondria is a very different structure to the chloroplast. So this is our mitochondria. In between here, we have our special structures. So we have our matrix. Mitochondrial matrix, the space in between each of these folds, and these folds have got a special name as well. We'll get to that later on. So the cells, this mitochondria converts the chemical energy stored in glucose into something that we can use. It converts that into ATP, or energy stored in ATP. We end up with 36 to 38 ATP molecules. So a considerable amount of energy. The happy thing with the synthesis of ATP is it's recyclable. So if we just have down here it's almost an ATP cycle really, but if our goal is ATP, we release energy, for whatever reason, we end up with ADP and an inorganic phosphate. And when we break down glucose using aerobic respiration, we're storing energy back into ATP again. So this is a perfectly recyclable process. Now obviously it's going to gradually break down, but ATP is incredibly resilient, and it's a really, really important mechanism, a really, really important tool for our energy use. Now this is all aerobic respiration. This is all when everything is working nicely. There's a whole nother topic or a whole nother issue with anaerobic respiration, which is where we have no oxygen or insufficient oxygen for the process. When we have insufficient oxygen, we're going to get less energy because less of the bonds can break down. And we'll deal with that later on. Where we're looking at the mitochondria now, these folds here become critical. So these cristae that make up the folds here, they are dotted with enzymes. Just like the endoplasmic reticulum is dotted with ribosomes and that increases surface area, the cristae are dotted with these enzymes. And the enzymes are really, really critical for this process of aerobic respiration. So we've just got our last little bit, which is a large central or vacuoles. So vacuoles are similar to vesicles. They are single membrane sacs that are used for long-term storage of food particles or water, enzymes, wastes, coloured pigments. In plants they have a large central vacuole, and this acts as storage as well as fulfilling a structural role in maintaining turgor. We've just got a very brief comparison between prokaryotes and eukaryotes on this last slide. So in one circular chromosome in prokaryotes, free in the cytoplasm, no organelles, and cell walls made up of sugars and proteins. In eukaryotes, we have linear chromosomes held within a nucleus. We have many organelles, and each organelle fulfills a specific purpose. And then lastly, a polysaccharide cell wall when there is a cell wall present, which, as we've talked about, is only present in plants and in fungi, not in animal cells. So that concludes the lesson for today. And you need to email me if you have any questions.